Hi, I'm Kate and I'm going to be conducting an experiment with the goal of taking the DNA out of a strawberry. For this experiment, we need water, isopropyl alcohol, salt, dish soap, a strawberry, tweezers, a small beaker, test tube, a strainer, and a plastic bag. Start by pouring 90 milliliters of water into a beaker, or in this case, a measuring cup, followed by 10 milliliters of dish soap and 1/4 teaspoon of salt. Stir the mixture. Place a strawberry in a Ziploc plastic bag, and then pour the previous mixture in the bag. Try and get as much air out and zip it closed. Begin to crush the strawberry into the smallest pieces possible, getting rid of any large chunks. After the strawberry is mostly broken up and mixed with the soapy water and salt, pour the contents through the strainer into a container. Pour this into a new container. Once in another container, add 5 milliliters of isopropyl and let the mixture aggregate until a thin white film appears at the top. This is the DNA of the strawberry. Now, let's find out how each of the ingredients plays a certain part in the collection of DNA and what their role is. DNA is normally soluble in water because the nucleotide bases are hydrophobic and they face inwards, and the sugar phosphate backbone of the molecule is hydrophilic and it faces outwards. The salt used in this experiment neutralizes the charge on the sugar phosphate and makes the entire molecule less hydrophilic because of the positively charged sodium ions neutralizing the phosphate anion groups on the nucleic acids. And now the DNA molecule will not dissolve as easily in water. Dielectric constant is the ability of a substance to store electrical energy in an electric field, and water has a high dielectric constant, so the sodium ions and the phosphate anion groups have a hard time joining together. Ethanol, on the other hand, has a very low dielectric constant, meaning that it is much more likely for the two ions to interact, making the nucleic acid more hydrophobic, separating from the water and easier to distinguish. Dish detergent contains a molecule called lauryl sulfate, which is used to remove hydrophobic substances such as leftover fats and proteins from dishes, and is almost doing the same thing in DNA extraction. The soap pulls apart lipids and proteins, making up the nucleus, protecting the DNA, allowing the DNA in the interior of cells to float freely, being released from the cell. This leads to DNA floating to the top of the mixture, visible for the human eye to see, and kind of looking like snot. That's all on how to extract DNA from a strawberry. Thanks for watching.